How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're listening to DNA Today, a multi-award-winning podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research. For over a decade, DNA Today has brought you the voices of leaders in genetics in over 200 episodes. For the past 3 years, DNA Today has won the People's Choice Best Science and Medicine Podcast Award. I'm Kira Deneen. I'm a certified genetic counselor and your host. This week, I attended Bio's CEO and Investor Conference in New York City. For those that haven't heard of the conference, it's one of the largest investor conferences in the biotech industry, where institutional investors, industry analysts, and senior executives come together to learn from each other and shape the future investment landscape of biotech. The organizer of the conference is Bio, which is a nonprofit policy and advocacy organization for the biotech industry. And during the conference, there were some fantastic panel discussions with these industry leaders. There were also company presentations where mostly CEOs would share what they're working on and include the stage of their research, what makes their company unique, and ultimately a pitch as to why investors should invest in their company. So after these presentations, I had the opportunity to catch up with a couple of the CEOs to learn more, which you're going to hear coming up, and wanted to thank Bio for having me attend as media and being able to share some of the highlights from the conference to you listeners. Our first guest is Dr. Premal Patel, a physician scientist with 15 years of experience in medical oncology drug development. I caught up with him after his presentation about how his company, Cellinfinity Bio, is using CAR-Ts to attack solid tumors, which, don't worry, he breaks down for us. He started by telling us about his education and clinical background that led to his involvement with Cellinfinity Bio.
Trained and got an MD and a PhD, so it's a dual degree program. Okay. And then I went through the clinical training and became a medical oncologist. So first there was internal medicine, and then there's medical oncology fellowship that was completed. But along the way, I mean, I've always had one leg in research and the other leg in the clinic. So even as I did the medical training, there was always that research side of me. And I've always been fascinated by both, really to advance medicine, as opposed to apply medicine, which is really fantastic. My wife does that. She's a physician. She applies medicine to people who have brain cancer, for example. But I've always been interested in, what can we do about these individuals at that next level, different therapies.
Yeah. So tell me about this company. It's a Cellinfinity. You did this great presentation and you have to go so fast here. So I'm glad I actually get to talk with you after your presentation. You're focused on CAR-Ts. So for people that don't know what that is, can you explain what CAR-Ts is before we get into how impressive the research and development that you're doing is?
Yeah. So over the last 10 to 15 years, we've really understood how to activate T cells to fight and many times cure cancer in a subset of individuals. So CAR-Ts is an example of that. Essentially what we do is take T cells, which are immune fighting cells that all of us have, take it out of the, isolate and take enough of them out of the bloodstream, and then re-engineer them with a warhead that will take the T cells into cancer cells. So we engineer them outside the body, grow them up in a petri dish, and then put many more T cells back into the human system. And those CAR-Ts float around the bloodstream. They identify cancer cells, bind to cancer cells, and then kill them. It's been very effective in liquid tumors. By that, I mean leukemias and lymphomas, resulting in about half the patients durable remission. And a good example is Emily Whitehead. She's a poster child. She's been cancer-free. And she was, in her before her teenage years, I believe, when she was first diagnosed with her cancer that was refractory to everything that she's been, but now she's been cancer-free post-college now, cancer-free over 10 years.
Wow. And she had a type of blood cancer.
Correct.
So exactly. It worked well there. You focus more on the solid tumor. So is this a newer area in terms of using CAR Ts to attack solid tumors? Do we not have that on the market currently?
Correct. So although when we put T cells back in the body, they can find with these liquid tumors in the bloodstream really efficiently. Solid tumors has been really challenging and
Hard to get to, I would imagine.
hard to get to, and it just doesn't work. And we don't fully understand what are all the complications. Why can't enough T cells get into solid tumors? But we do feel that we understand some elements of it. One is it has to actually invade the solid tumor. T cells have to go from the bloodstream into the solid tumor environment. And solid tumors are things like, brain cancer, prostate cancer, breast cancer, lung cancer, stomach
Anything
cancer.
that's a specific mass where that started.
Exactly. And so the T cells have to go from the bloodstream into these masses, and they don't do that very well. And then the masses themselves, if you think of it as a ball of cancer, once T cells go in, they're sort of lonely and they're around a very hostile environment. And that hostile environment quickly rejects those T cells.
Where the cancer might keep growing and proliferate, whereas the T cells not so much. And that's what we want. We want the army to be going and fighting for us of those T cells. So when you mentioned engineering and adapting our T cells and then putting it back into the person's body, so is that genetic engineering that you're doing? What are you doing at that point in the process?
That is so. And then we took the premise that we don't exactly know what genes would be really important to get the T cells into a solid tumor environment or that mass. And then once it's there, does it divide and kill? We don't know those genes, right? Others have tried rational approaches, but we took a very empiric approach. So the first thing we did is we created a large pool of cells, but each cell is different by one gene. And we have 20,000 genes in our human system. So each one of the genes can be altered. We can knock out each one individually. So each cell harbors, in an ideal scenario, one alteration. So I describe this as either a marathon race or an Olympic race where we have 20,000 participants. And then you ask, without knowing what gene does what, you devise a competition. So the competition might be which of these 20,000 cells best kills the solid tumor, best enters the solid tumor mass, right?
Right, because first you just have to get there.
Exactly. So you could you could run all these races. So one could be getting there. The other one could be, does it work in this? You create different hostile environments. Another could be, does it persist? And by that, I mean, does it kill not only for a day or two, but kills for weeks to months, continues to kill tumor cells for many weeks to months. So these are different races or competitions that we put our T cells in. And then we identify, okay, what are some of the genes that are involved in each of these important elements that we think we need to ultimately confer. So the other way of thinking about this, if as a program person, if you're a computational person, we need to reprogram our genetic code so that we allow the T cells to harbor some functions that it doesn't currently have.
Yeah, boost it more.
Exactly.
And with that, you're looking at, okay, which genes that you're either knocking out or whatever, which ones are going to work to get the CAR Ts to the solid tumor, but then which are going to stick around and actually attack it. So it's like the step one, step two.
Step one. So each of these steps are, when we do this in the lab, each one is individual. But then the second step would be, okay, what if so gene A is involved in this very important property, but gene C is involved in another property. Then we combine gene A plus C. And
right. So you have to see, well, what mixture is going to work the best. So to figure all of those genetics out, then are you using something like MAVES to figure out, because you have to run a lot of tests at once if you're looking at theoretically all the genes, like 20,000.
But you could also select. So for example, if it's a winner takes all and the losers die, which is how we typically run our experiments.
It's cutthroat in the lab.
Exactly. It's cutthroat, exactly. So yeah, and that's generally how we design our experiments. But you're right, if we wanted to test each one individually in a well, then we'd need 20,000 wells and test each one individually. A faster way to say is only the winner wins, right? Only the winner survives.
Okay.
And that's
To narrow it down.
exactly.
Yeah. Interesting.
Or the top 100 winners, right? Essentially.
Sure. Cellinfinity, you've been able to optimize CAR Ts to actually be able to do some of this. And was it focused on renal cancer? Is that what you were focused on?
Correct. So our target, the warhead ultimately that we engineer is, and would allow the T cells to bind to kidney cancer, right? And that's one aspect of it. But then we also reprogram the genetic code so that once it binds, it actually enters and does the things that it needs to kill some kidney cancer.
Interesting. And you've done this in mouse models, and it's the results have been pretty good.
Exactly. Yeah. The mice models, tumors go away. And yeah, it's very important to say that this is working in preclinical. So we haven't done clinical studies yet. And that's next for us, obviously.
Right. And that's part of being at this conference, is having the opportunity to present your company. And this is what we're doing, because we're looking at these companies that are presenting. I'm here to just observe and share it with the masses, but hoping for investors to become interested and do investments. So what would be the next steps if there was an investor, especially if there's one listening right now that's really intrigued by this and is like, oh, I want to look at Cellinfinity in terms of it having their own investment? What would you do with their money? Why you?
Right. So, it's essentially research takes funding, clinical trials takes funding, and the funds that we that investors give us would be used directly for the programs and doing doing the clinical studies. Now, why us, right? So this one challenge of coming up with the best CAR T, what I call, the best in field product, best in class product. I think we have that because we've been able to gauge against our competitors, and ours do work really well. The second, which we haven't talked about, is right now that engineering is all done outside the body, right?
Yes, that was a big focus.
So the technology that we're developing to complement is to do this all inside the body. So essentially what we take is instead of doing all these manipulations and genetically reprogramming in a petri dish, what we do is give a virus, which would go into a T cell directly.
So it would be like the delivery model to put the information into the virus, and then you could give that to a person instead of taking their T cells out and putting them back in.
Correct. And the virus goes to the T cells and then introduces a genetic element and reprograms the T cells. So this is a really new technology, and there isn't any clinical data that I know of.
And you would know, Dr. Patel, yes.
They're just now starting clinical studies with some of the first out of the gate companies. So stay tuned. I think this will be really, and the reason why the field across the board is really excited by this approach is now it makes this form of gene engineering CAR T accessible to many, many individuals, right? We don't have to wait around for taking T cells out, engineering them, expanding them, putting them back into individuals. That's long, expensive.
I would think weeks, months.
Weeks, exactly.
Once it would get to a clinical trial phase of just how long that would take.
Yeah, and individuals may not have that long, especially with terminal cancers. So we need to be much faster, but also much more economical. I was born in India, so one of the things I'd love to have is therapies that the entire world could access in terms of cell and gene therapy. So might this be, as expensive or not as expensive as an antibody-based therapy ultimately?
Yeah, and you had some in your presentation similarities to. We're at the point where it's something you said was similar to antibodies. What was that part?
So the overall development cost, right, is now for example, the ex vivo approach costs $400,000 just per patient to do that, right?
$1 million almost.
Yeah, exactly. And then antibodies per dose could be a few $1,000s or less, depending on the antibody. So we want to get it closer to a few $1,000 range and not hundreds of thousands of dollars, right? That's our goal.
That's so exciting. Well, I wish you the best of luck. And we'll put your contact information into the show notes of this episode if people are interested to learn more. And thank you so much, Dr. Patel.
Fantastic. Thank you.
Our second guest is Dr. Travis Whitfill, who has a background in molecular biology and biochemistry after receiving scientific training at the MD Anderson Cancer Center, Duke University, and Yale University. Dr. Whitfill is the co-founder and chief operating officer at Azitra, which is what we chatted about. So Dr. Whitfill, you are a serial entrepreneur, so this is not your first company. What companies have you been involved in before? What is your background in?
Yeah, so I actually spent 10 years in venture capital.
Okay, so you know the other side of it, which is really helpful.
Oh, yeah. And I also am an adjunct professor too. So I've been on, the research side and the investing side, and especially the entrepreneurial side. So yeah, I spent 10 years in venture capital as a partner in a fund called Bios Partners, building really early stage biotech companies. So I helped start Actuate Therapeutics, which has now grown to, phase 2 clinical trials in pancreatic cancer. I'm on the board of Innate Bio, which is a company that's developing cell therapy for oncology, and a number of different other companies from oncology to ophthalmology and kind of everything in between. So
wow, so you're bringing a lot to the table just in terms of this company and everything that you're doing.
I've seen a lot.
So what led you to actually co-found Azitra? What need did you see to say, I need to start this company because I want to fix this?
Yeah, so this was in 2014. I was a grad student at Yale, and my co-founder, Azim, that's how we got the name of the company, is...
Fun fact there.
Yeah, using our names together. We were really interested in synthetic biology, and we were seeing people start these fecal microbiome transplants that were very effective in treating C. diff infections. And then we saw people trying to engineer gut bacteria to deliver things into the gut. We thought that was kind of crowded, and it's really hard to see an effect in a gut disease. So we were talking to a lot of immunologists and different people around the university, and found that the skin would actually be a really great organ for this approach. We talked to a lot of dermatologists and got a sense of what they're seeing. And there's a lot of skin diseases that are really big pain points that they don't have an approved treatment option, and actually have a really bad quality of life in a lot of people. So we decided to look for dermatology indications that we thought were interesting, and found that there's a lot of genetic skin diseases where there's a simple genetic component that causes a missing protein or a misfolded protein, and that leads to really bad consequences. I recently went to a conference last year with an ichthyosis population of people that have genetic diseases of the skin. Some of them are in wheelchairs. Some of them get recurrent skin infections. One patient had to get his hip, a 23-year-old had to get his hip replaced.
Oh my God.
Because he was on steroids for so long. So a lot of these patients have really big problems that are really under addressed.
And it's interesting that you kind of started by saying that the gut microbiome, you're like, well, that's kind of a crowded space. When most people think about the microbiome, I think they are thinking of the gut, and they're thinking of all the different bacteria strains that we are walking around with. And not many people think of, the skin microbiome. Maybe people that have a really intense skincare routine, which I could probably learn from. But it's interesting that you saw this area to go into. And like you said, that some skin conditions are like monogenic, caused by one gene. And so that's a little bit more straightforward than some other conditions that are caused by many different genes. And there was one that in your presentation you were talking about that is a rare disease, and it is Rare Disease Month. So I figured I had to ask you about this one, Netherton syndrome. So tell me about this condition and how the genetic variant then leads to, it's a certain protein that's not being produced enough, or is it's being produced, but it's not folded right? What's the problem there?
Yeah, usually there's a mutation. So the gene is called SPINK5, and it makes a protein called lectin, which is a protease inhibitor in the skin. So a lot of these patients, there's been 80 different mutations identified in the gene that cause Netherton syndrome. So it's pretty heterogeneous in terms of the different mutations, but it leads to some kind of usually lower expression, no expression, or some kind of misfolding where it loses its functionality. But in all of those patients, when you don't have this protease inhibitor, you get the result of these overactive enzymes that chew up the skin, which is a huge problem. And that leads to the manifestation of the disease, which is these scales, redness, inflamed skin, and then, basically no skin barrier to protect from pathogens and things like that from causing infections.
So not only is it sounds like a painful condition, but also exposing your skin is a barrier we really need, and it protects us. And it's part of, I suppose, our immune system in that way. So if you're not having that barrier functioning properly, that our organ of our skin functioning right, we're more susceptible to that.
Yeah, exactly. And very sadly, this is not just something that really affects kids. It really affects throughout their whole life. I mean, I know 2 patients that were in their 30s that died from sepsis because they got a skin infection that was uncontrolled and didn't get care fast enough. So it can be a deadly disease because of that.
Yeah, you even mentioned that 10% of infants with the condition also pass away from it. So it is a really serious condition,
It is.
as unfortunately a lot of rare diseases are. And there's no treatment specifically for Netherton syndrome. This would be if this passes FDA and all the other steps you have to, go to get there. Sure. If we see it go to market, it would be the only one on the market, presumably.
That's right. Yeah. Assuming others don't get to the market. But yeah, the point is there's nothing approved. And the only thing for these patients to do is to take oatmeal baths or put on moisturizer. And that doesn't do anything to rebuild the skin barrier. So
that's not a treatment. That's just something to help in the moment, if
exactly.
anything.
Temporary relief.
That is the ATR12 candidate. So the other one that I was intrigued by is the ATR, is it 04 candidate?
That's right.
Okay. Yeah. So tell me about that, because our audience might be familiar with EGFR and how that is often targeted for cancer therapies, looking at that gene. But what I learned in your presentation is that people that are on this drug can often have a rash associated with it. Right. So there's a whole nother candidate that you're developing to treat that, which is also genetic related in that sense.
Yeah, exactly. Yeah. So these patients that are undergoing EGFR inhibitors, which is a targeted therapy for a specific mutation in cancer, the drugs work really well in treating the cancer, but they have this really bad side effect of causing a skin rash, because it suppresses your skin's immune system and leads to inflamed skin. It's usually around the face, in upper back and upper chest. But as many as 10 to 15% of these patients will stop their cancer treatment because of the skin rash.
That's a reason to stop treatment, because the rash is that bad.
So bad, exactly.
Wow.
So they literally would rather have cancer than this.
That's saying something.
Yeah.
And that's a very common, just the fact that I've heard of it. I don't work in oncology. Mm-hmm. It's common enough that it's not just one type of cancer that has this type of drug. It's a long list.
Exactly. Right. And it's not just EGFR inhibitors. There's a lot of other targeted therapies that have a similar side effect.
Okay.
And the current standard of care is, high dose antibiotics and steroids, which if you're doing that for a year, it's really bad. I mean, it can cause more problems. So
that can mess with your gut microbiome.
That's right.
Exactly. Full circle. But it's interesting because you talked about how the cancer drug can suppress the immune system.
Right.
It's causing inflammation and also a certain bacteria to grow. And so this drug that you're developing would be not just looking at one of those, but really tackling both of those issues if it works. Right?
Exactly. Yeah.
A lot of, CYA statements going on.
Sure. I will say, in our preclinical models, we saw 2 things: a reduction in inflammation measured by some specific biomarkers in the skin, and 2, one of the major pathogens in this rash is Staph aureus. So we saw that our candidate reduces Staph aureus levels by 99% in the skin that we saw. So that's our approach for this is kind of the dual approach of Staph aureus and this inflammatory marker reduction.
And it seems like you are the only one on the market in a lot of ways that there are elements that other companies have separately, but you have all together. So what if there are investors listening, because we're at a conference where we're hoping a lot of these companies will, woo investors, say, yes, we want to invest in this company. Why Azitra? why should they invest in your company over others or people that are in the same space as you?
Sure. I think two things. First of all, we're going after indications where there's nothing approved. And I will say we have a third program under development that's for ichthyosis vulgaris, which is another genetic skin disease caused by missing filaggrin. So in all of those diseases that we are addressing, there's nothing approved. So the dermatology space has gotten really crowded in things like atopic dermatitis, psoriasis, alopecia even. So that we're trying to stay away from really crowded diseases and go after things where there's nothing approved. And it's much less crowded. There's maybe one or two companies working in the space. And I think we're also really differentiated because we're a disease modifying approach. No other company has been able to achieve this, basically protein delivery system that we've developed, because it's very hard to penetrate the skin barrier and deliver large molecules to the skin. But by using skin bacteria that are naturally found on the skin, they localize to all the different tissue layers, even down to the hair follicles, where there's an interaction with the immune system, for example. So we solve that problem of the skin barrier.
And that's just through an ointment.
Yeah,
like
exactly.
That's as simple as it is.
Right.
Which in terms of the delivery model, but in terms of the back end of how you got there is talking about isolating plasmids and having this going. So a lot of interesting, would genetic engineering be the right term?
Yeah,
for
absolutely.
That? Okay. Yeah.
Yeah. And
I think of like CRISPR and different things, but there's a whole lot within the genetic engineering space.
Yeah. So what we do is we integrate the gene of the protein that we're expressing into the chromosome of staph epidermidis. So it's integrated into the genome of staph epidermidis.
So that then that bacteria just starts making the protein that the person is missing.
Exactly. And then we've also attached this signal to the protein that the cell recognizes, and it immediately exports the protein out of the cell. So as soon as it's made, it pops out. So we're able to leverage the innate, or I guess like the natural biology of bacteria that has these secretion signals, so we can secrete the protein. And then the final piece of the genetic engineering side is that we've also made all of our candidates to be oxytrophic. So
what does that mean?
Yeah, so we've deleted 3 different genes in each of the strains that are responsible for making a certain amino acid that is critical for cell wall synthesis. So what we've done is we have to, in our formulation, we add this missing amino acid that's synthetic and not found in nature, put that together, and the bacteria is able to survive and make the cell wall. As soon as that synthetic amino acid, is called dealanine, is used up, the bacteria die. So that way we can control the growth. And if you were to like shake hands with someone on the street or whatever, you're not going to spread the live bacteria.
Like if you've applied the ointment to your skin and then you're shaking someone's hand, you mean in that sense?
Exactly.
Okay.
Right.
Yeah. Interesting.
Yeah.
Well, thank you so much. I know it's a busy conference, so I appreciate just being able to sit down and chat with you about all of this. We'll make sure to put your contact info into the show notes for maybe some investors that are listening that are interested.
Wonderful. Thanks so much. Appreciate it.
For more information about today's episode, visit DNAtoday.com, where you can also stream all 200 plus episodes of the show, including video versions of interviews recorded in 2021 or later. Any questions, episode ideas, guest pitches, or sponsor inquiries can be sent into info@DNAtoday.com. Be sure to follow us on social media, especially so you don't miss a giveaway. We are at DNA Today Podcast on Twitter, Instagram, LinkedIn, Facebook, and more. Please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. Here's the bonus. If you do and send us a screenshot, I'll give you a shout out on the show. DNA Today is created, hosted, and produced by myself, Kira Deneen. Our team includes communications lead, Corinne Merlino, video lead, Amanda Andrioli, outreach intern, Sonia Tanaiker, social media intern, Kajal Patel, and graphic designer, Ashlyn Anokian. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and me, are all made of DNA. We're all made of the same chemical DNA.